It's 4:20, so let's talk about the lab. Okay. Now maybe, I think this week, it's more helpful. Maybe in the future it wouldn't be, but as helpful. But to have the lab in front of you when I do this. So I'm gonna go ahead. Well, then I gotta get my explanation. Maybe not. Dang. Okay. Ignore what I'm saying. Okay. So this week's lab, it's on the scientific method. And I think this mini lecture is going to be shorter. I'm just going to kind of go through another example, really, like we did in lecture, and talk about just kind of what's included in this lab, what you'll be doing, but that shouldn't take too long. And that's it. So let's go ahead and do one more example, because this is what you're going to be doing in the lab. You're going to be forming your own observation, question, hypothesis, experimental idea, and then even testing one that you come up with at home. It was our department chair's idea. I think it's kind of cool. I'm excited to look and see what you guys come up with. For now, this always keeps sliding down. Let's do one more example here of the scientific method and going through it. So let's say our observation is that students earn different exam scores. Students earn different exam scores. Again, the observation is typically a problem you're looking to solve in the world, or improve. A technology you might want to improve, or like I said, a problem like maybe global temperatures are rising. You know, the virus one I mentioned in lecture. Things we're looking to solve using science. So students earn different exam scores. We want you guys to all do well on exams, right? You're gonna want to solve that. So we ask the question, how can students, question could be, how can students earn a high exam score, or higher exam scores, a high exam score. How can students earn a high exam score? So that's our question. So we can obviously formulate many hypotheses. We could say students who study a lot can earn a high exam score. We could say students who have a previous background in science earn higher exam scores. Students who exercise more, or what would be another one, or another reason? Students who are genetically predisposed to be better in science than they are in English. Plenty of things we can come up with. My example is about office hours, so let's do that. So the hypothesis could be students who attend office hours earn high exam scores. Okay, so based on our lecture today and what we learned about independent, dependent variables, what's our independent variable for this hypothesis? What are we, the researcher, introducing and interested in seeing will influence our dependent variable? Yeah, the office hours, totally. So that's our independent variable. So what variable, or what's going to depend on whether they go to office hours? What directly depends on it? Yeah, their exam scores. So that's our dependent variable. The high exam scores. Awesome. Okay. So I'm going to erase the top here and keep that up. So now I like this example because they're like the plan example, there's so many different examples of controlled variables you can come up with. Controlled variables. In the lab, it calls them confounding variables. Same thing, same thing. It mentions or controlled, same term. So controlled variables. I realize that red, this is, my other red is not good. I need to get better red markers. Well, let me scoot it up. Okay. So for this hypothesis, if we want to directly see if the office hours influence their exam scores, we're going to have to control for a lot of things, such as the amount that students are sleeping. So remember, our controlled variables are anything that could influence our dependent variable here, the exam scores, other than our independent variable. That might give us an inaccurate conclusion. So sleep could do that. Students who don't sleep as much, but maybe they went to office hours, might get poorer exam scores. And it might lead us to thinking, oh, the office hours aren't helping, but it's because they didn't get much sleep. Age, sure. I think that's a good one. Oh, I do have that on my list. Yeah. So maybe they have more life experience and they could take better exams in that sense. Sure. Oh, people are coming up with a lot of examples. Time spent studying, totally. That's a great one. Because that's not our independent variable. Drug, Adderall, yeah, totally. Oh, these are great. Coffee use, Adderall. I mean, there are so many you can come up with. Coffee, Adderall, whatever, stimulants, I guess. study habits. What else do I have? I think I have diet. I'll just list one more because there's just so many. That's the problem with human studies. I feel bad for a lot of people in psychology and those neuroscience that are working with humans, not mice. Things that are mice, you can really easily control their environment. But let me see. What else do I have? So I'll write their scientific background. Scientific background. But there's tons, right? You kind of have to control these people's lives in a sense to do an accurate study. Okay, so those are some great examples. Let's continue on in our scientific method. So the next thing we have is the experiment. Now that we have our hypothesis, we have to conduct the experiment. And the experiment is, oh, people in the waiting room, get them. The experiment, we're gonna collect exam scores. And since we want the experimental group and a control group, we're gonna collect exam scores from students who attended office hours, students who attend office hours, and we're going to collect exam scores from students who don't, and from students who do not. Oh, I just saw in the chat, methods of studying. That's another great one. How are they studying, if they're studying? Totally. Maybe someone's studying with the TV on. Totally. Okay. So now that we have the hypothesis written down, let's continue here. And first, let's identify from the experiment my experimental group and my control group. So the experimental group will be the students who attend office hours. That's our experimental group, right? And then our control group, remember a control group, we remove the independent variable, which we said was the office hours. Those are going to be the students who don't. This is our control group. Okay, next step in our scientific method, it's our analysis or our results step after we collect our data. So this is the analysis / results. Accidentally sort of got in the way there, so I'm gonna write it down here. So what we're going to do is we're going to have our graph in our results section. So we're graphing our data, and we're going to look for potential relationship between office hour attendance and exam scores. So we're going to analyze the data. Look for potential relationship between office hour attendance and exam scores. We want to see if our hypothesis is supported by our data. So we're going to analyze it with a graph. Potential relationship between office hour attendance and exam scores. Okay, and then we just have our last step. Trying to think, I'll write up here. Usually in class, I have a giant whiteboard, so don't really run out of space so easily, but let me. So last step is the conclusion. Sometimes scientific papers will have a discussion section as well, but the typical next step is our conclusion. What do our results tell us? And let's just say, for our example, that the hypothesis is valid and that the data support the hypothesis. In other words, attending office hours increases students' grades or something. So the data support the hypothesis. So this lab is actually pretty straightforward. You guys will be doing practice problems, really going through the scientific method and reading about different types of graphs. So I mentioned a graph that would be used to visually represent our data. You will be, Sophia. So Sophia asked if you'll be emailed the lab packet. And yes, so after lecture on Tuesday and the lab lecture, I email out the packet and any links or any information to you guys. Okay. So graphs are in the lab packet. So it just explains what kind of graph you would use depending on the kind of data you're generating in your experiment. And it explains a lot about, in fact, I'll talk about it, why not? Depending on the kind of data, that's going to directly affect what kind of graph you're going to use to represent or show your data. So the first couple pages, you're going to be doing some practice, identifying, like we did in the lecture, and like we did here, independent variable, dependent variable. examples of controlled variables or confounding variables that we listed for different hypotheses. Let's see, and then, let's see, blah, blah, blah. Okay, designing your own experiment. So one of them is designing your own experiment, your own hypothesis, but not actually going through with it. And then there's one where she does want you to do it and collect data at home. So you could do some interesting like things related to cooking or cleaning, or even like something with plants. I've seen a lot throughout my time as a teacher, but be creative. Let's see. So let's talk about graphs. So let me go ahead and erase this. Oops. Okay. So there's three kinds of graphs that you'll encounter in science. There's what we have a line graph. And a line graph will visually show continuous data. And really almost always, when you see a line graph, it's data that takes place over time. And so the example shown here, candy sold by a third grade class in a number of weeks. So there's your line graph. So really, line graphs are almost always going to be continuous data set over time. The much more common graph you see is a bar graph. It's actually certainly a graph that you'll be doing in this packet. And a bar graph's used for showing distinct data points between two or more categories. So we sometimes, as opposed to continuous data, this is categorical data. Two or more categories. So this is in your lab packet, the example is a favorite dog breed survey. So there's different categories of dog, right? Different types of dogs that are represented on the x-axis on the bottom there. And then the number of votes is represented on the y-axis on the vertical axis. And the last kind of graph that you'll encounter is a scatter plot. Oh, I will, well, maybe I mentioned that in the bar graph, it is distinct data. It's not continuous by any means. Like continuous data you're measuring over time is just, it's continuous. But a distinct thing like the number of votes for a favorite dog, you can't have like 4.5 votes, right? It's a distinct thing. It's not continuous. So that's worth mentioning. A scatter plot shows a potential relationship between two variables. So actually, what example did they use? Internet usage. Oh, that's interesting. So they, the example in your lab packet is internet usage and age. So the internet usage in hours per week is there on the x-axis or the horizontal axis there. And then the age is on the vertical axis. So you'll see all these points, and it's a scatter plot showing a potential relationship between how much you use the internet and how old you are. So scatter plots are used to show a potential correlation or relationship between two variables like that. I would say that, like for the hypothesis example we did, it's more categorical because it's those who attended office hours and those who did not. And then you would have maybe average exam score on the vertical axis, something like that. So it's a much more of a categorical type thing. So speaking of graphs, I will mention, so let's see what page it's on. So you might want to write down so on page 9. So I'm going to send this to you guys so you'll know exactly what I'm talking about. But for now, just write down page 9. It's at the top, and it's this part B. Okay. You're going to be making a graph. And the graph is going to be, so remember last week for the metric system lab and measuring, Stacy had in the data table, oh, I should have had it out. He had some class averages of percent error. So let me get that out really quick. Okay, so mine's blank, so ignore mine. But for last week's lab packet, Stacy had some numbers here. And this was the class average for percent errors, depending on the measuring device. Is this ringing a bell? So when you would use each of these to measure the amount of milliliters and calculating your percent errors, Stacy had some. Normally in class, we'd have our class averages. I would collect them, but you're going to graph them in this week's lab. So that's going to be on page of this week's lab. It's page 9 again, and it's that B at the top. Those are my notes to myself. And like I said, the best graph to do for that would be a bar graph because it's categorical data. You have like those 4 categories whether you're, let me see. So you're doing the, so transfer pipette, right? The, what is it, 25 milliliter graduated cylinder, 250, 50 milliliter beaker, and a transfer pipette. So you'd have those 4 categories on the bottom, and then along the side, the y-axis, you would have the percent error that you calculated, or that Stacy would have listed. So that's just, it's a practicing making a graph. Yes, so that's the only data from last week's lab that you'll use in this lab. So that brings me to my next point that I really want to make sure I tell you guys. So, okay, yeah, this goes here. Okay. Last week I didn't have you guys measure, right? Remember I told you to cross out that thing where it says measure your forearm length, height, all that stuff. Okay, so because of that, we're not going to do some parts of this lab, but that's okay because it's just practicing with a scatter plot, and it gets too crazy because I'd have to get all of your data. And so let me tell you what you're not doing. You're still going to be practicing graphing. Trust me, you're practicing graphing the percent error, just what I just mentioned, and also your own data that you'll collect during the lab. But write down that when you get the packet, not to do you'll do the pre-lab, you'll do everything except page 8, part A. So I wrote, do not do this, page 8, part A here, and then this as well below it. So this is on page 8 of the lab packet. So just write that in your notes so that when I send it to you, you'll know don't do this. Page 8. It's really all of page 8 here. What time is it? Okay, I'm okay. But you will be doing the next page. That's the percent error graph. Like I said, a bar graph would be best for that because it's different categories, what measuring device it was. Again, with graphs, oh, yes, I will repeat that. So page 8, that's correct. You won't be doing that. And that's for you guys, the synchronous section and the asynchronous section that I send this to as well. Both of you all not. Okay, so just want to mention again, it's sort of included in my instructions, but whenever you do a graph, unless you're savvy enough, you want to do it in Excel, totally okay. Stacy included a link, where is it? It's actually on page 8. If you want to use what's Google Sheets, which is an online way to make a graph, she has a link for a tutorial. Up to you, you can do that. And that'll show you how to do it with Google Sheets. You can also just hand draw the graph and use one of the apps I recommend, Adobe Scan, Evernote, and upload it as a separate PDF or however you want to do it that way. Totally up to you. So I wanted to mention that. And what else? I think there's one more thing. Like I said, you're going to be designing your own experiment. So I'm curious what you guys will come up with. That'll be cool. Because usually in class, we don't do that, right? Can't really do an experiment at home. Okay, because the post-lab question has to do with measuring the arm and everything that we would have done in class, where we have a measuring meter stick and everything, you won't be doing that. So that's the only other thing you're not doing, which is the last page, 12, the only post-lab questions. You won't be doing that either. But everything else you will be, except, what was it, page 8? Let's see. Yeah, page 8. Page 8, and then this. That's all you won't be doing. Page 8 does have the link on it for Google Sheets if you want to do it that way. But that's it. That's it. It's just, again, practicing what we went through today and just now, doing some graphs. Side note, the independent variable is always on the x-axis. But again, the packet and Stacy's videos, she talks about this. The y-axis will have your dependent variable. That's how we observe our data with these types of graphs and what we collect for the dependent variable. So are there any questions? I want to let you guys go because you're probably sick of me at this point. Any questions? I'll be sending this out to you guys shortly with today's recording, of course, as well. Sending you guys a Microsoft Word version, Google Doc version. Cool beans, love it. Okay, great. Well, enjoy the rest of your evening. It's beautiful out. And I'll see you guys Thursday. All right. As always, email me with any questions you have. Thank you. Appreciate it. All right. Bye, everyone.